Cholesterol. Structurally, cholesterol has 4 hydrocarbon rings, a hydrocarbon tail and a hydroxyl group. 20% of the body's cholesterol comes from the diet, while the other 80% is synthesized in the body. Cholesterol synthesis mainly takes place in the liver and consists of 3 major stages. During stage 1, there are a number of reactions to convert acetyl-CoA into isopentyl pyrophosphate. This uses the HMG-CoAs reductase enzyme. During stage 2, the 6 isopentyl pyrophosphate molecules are condensed to form squalene. And finally, in stage 3, squalene is folded and sealed closed to produce cholesterol. Cholesterol is an important component in a number of body processes. For example, it makes up 25% of cell membranes and works to break up the van der Waals forces between the phospholipids. This leads to a more fluid and therefore dynamic membrane. Cholesterol is also used to synthesize a huge number of steroid hormones. These all have the characteristic 4 hydrocarbon rings and include well-known hormones such as testosterone and progesterone. Going back to the structure of cholesterol, having this structure means cholesterol is amphipathic. This means it has both a hydrophobic region and a hydrophilic region. Although it does have a hydrophilic region, cholesterol will still not dissolve in the blood. It therefore uses lipoproteins as a carrier. There are a huge number of different types of lipoproteins with varying densities. The main carrier of cholesterol is the low-density lipoprotein, also known as LDL. In the blood vessel, LDL binds to specific LDL receptors. These receptors then allow for the cholesterol to be absorbed into the tissues. LDL is often referred to as bad cholesterol. This is because high levels of LDL in the blood can lead to the accumulation of LDL in the blood vessel walls. These can then lead to the formation of hard plaques. With the addition of more LDL, these plaques can become large enough to stop blood flow completely. Atherosclerosis, which is the condition in which the arteries are blocked due to these plaques, can lead to heart attacks if the blockage is in the heart, or if the blockage is in the brain it can lead to a stroke. However, if we go back to our lipoproteins, high-density lipoprotein, or HDL, in contrast to LDL, is dubbed good cholesterol. This is because it works to pick up loose cholesterol in the body, reducing the buildup of cholesterol in the blood. As we saw previously, LDL is the main carrier of cholesterol. This means that atherosclerosis and therefore heart attacks and strokes are still very common. So what are the main ways to stop cholesterol building up in the blood vessels? Firstly, the amount of cholesterol in the diet can be reduced. Cholesterol is particularly high in foods containing saturated fats and trans fatty acids. However, if this is not sufficient, there are 2 main clinical methods for further reducing cholesterol. The first is to regulate bile salts. Bile salts signal for the absorption of cholesterol from dietary sources. By increasing the amount of bile salts that are excreted from the body, there are then fewer signals for absorption of cholesterol. This can be done using drugs such as cholestyramine. The second method is to reduce the amount of cholesterol synthesis. This can be done by inhibiting the HMG-CoAs reductase enzyme, which is essential for the first stage of cholesterol synthesis. This can be done by using a class of compounds called statins, which include drugs such as lovastatin. So there we have it. Cholesterol, not that hard to learn from a textbook.